right, everybody. Welcome to another Expert Talk. This is your extended family, and it's your time to share with us after our speaker. And just always realize how important you are, because without you, we wouldn't be here with the purpose that we love. And with that, I'll turn it over to you, Indu.
Thank you. Thank you, everybody, for joining today. And I'm really excited to actually introduce Dr. Suma Babu, and I'm really excited to have her talk to us. This is our first time actually meeting all of you. So, is co-director of Neurological Clinical Research Institute at Massachusetts General Brigham, and Dr. Babu serves as a associate professor of neurology at Harvard Medical School. Suma's research is all centered around improving outcomes for people with ALS. And she's passionate about therapeutic development for ALS. She has a special interest in early phase clinical trials and gene targeted trials in ALS. Her studies are being funded by NIH, PCORI, and industry or foundations. Dr. Babu is a PI for Healey ALS MyMatch program, which is the topic we're going to talk about today, launched in 2025, which is a series of patient-centric, multi-site phase 1B, 2A biomarker-driven clinical trials. She leads the neuroimaging research program for ALS at Massachusetts General Hospital and co-chairs the NEALS Neuroimaging Subcommittee. And really excited to have Suma Babu here, and take it away, Suma.
Thank you so much, Indo. This is very, very exciting to meet you all today evening, and thank you for making the time for today's talk and today's meeting. I'm excited to share a new program that we launched this year called the Healey ALS MyMatch. With that, let me share my screen. Right? I hope everybody can see all the slides. Great.
Yes.
Yes.
Right. And this is a new program that was launched in spring of 2025 by the Healey Center for ALS at Mass General Brigham. And many of you are familiar with the Healey ALS platform trial. And just to show a schema of where platform trials sort of is positioned in the entire drug development pipeline, and how we are positioning the Healey ALS MyMatch program in relation to the Healey ALS platform trial. So the platform trial is a later phase clinical trial program that's looking at clinical efficacy testing to understand whether a drug has a clinical benefit or not. Now, prior to answering the question of whether a drug has clinical benefit or not, there are numerous other questions of scientific importance that need to be answered in the earlier phases of clinical testing. A is to understand whether the drug is doing what it's supposed to do or what's hypothesized to do. Is it engaging with the target that it is hypothesized to engage with the target, or a protein in the brain or the spinal cord or in the peripheral nerves or in the muscles, whatever may be the target. Are we using the right dose for testing in ALS to understand whether it will have a benefit or not? Is it crossing the blood-brain barrier? It is entering, if you give it by IV, you give it by mouth, give it by subcutaneous injection, does it reach the brain? Does it reach the motor neurons? And is it safe and is it tolerable to take it over an extended period of time? These are types of questions that we want to answer even before answering the question of whether there is a clinical benefit in slowing the disease progression. And to answer those questions, we've positioned the Healey ALS MyMatch trial program. And this falls under the category of what Indu said earlier as 1B/2A. This is all of what I said before, sort of summarizes that particular phase of testing. And then phase 1A is more sort of first in human studies, that's generally tested in healthy volunteers. And sometimes it can be tested even in ALS individuals before moving into a later phase of testing. And then the final phase of testing is a phase 3 before it goes into to the FDA for as a new drug application for potentially for FDA approval. And this is almost like a funnel. We start with a large number of drugs in preclinical phases of testing. And as you go through the pipeline systematically, we realize that some of the drugs don't work. They're not safe. They're not doing what they're supposed to do. And then the funnel becomes narrower and narrower. And ultimately, what crosses the finish line for FDA approval are very few drugs, as we know, from ALS as well as every other disease for that matter. The Healey ALS MyMatch, where its position is so critical in terms of really keeping the ball rolling in terms of drug development and understanding what are those promising candidates that can move forward into clinical efficacy testing. So we're building this Healey ALS MyMatch on two guiding principles. One is it's all about the speed and efficiency. How quickly can we understand the key scientific questions in earlier phases of development, and how efficient can we be with time, with resources, and available participants to participate in these trials. And while doing that, while we're building on speed and efficiency, we also always want to anchor on the fact that we want to generate value-based information from this phase of testing before we go into the later phase of understanding clinical efficacy. So speed, efficiency, and value from drug development are our guiding principles for ALS MyMatch. Now, why is this important? As I showed that big funnel where a large number of drugs that are entering into the clinical phases of testing, and this is a very fluid space. Sometimes many drugs are in the later phases of testing. For example, when the Healey platform trial was launched back in 2020, many drugs were in the later phases of testing. But now the paradigm has shifted more towards the earlier phases of testing. More and more drugs are entering into the early phases of testing, or they are currently in early phases of testing. And it's creating a pipeline pressure where we have more than 100 drugs that are in earlier phases of clinical trials, and we need to test them in a very efficient way. We need to find out what drugs are promising that we can then move forward. And how do we then think of this pipeline pressure and the problems that this pipeline pressure actually generates, and how do we tackle those in early phases of testing? That segues into the first and foremost question of how do we match the right people to the right trials based on disease biology. We all know that ALS is such a heterogeneous disease. If you've met one individual with ALS, you've only met one individual with ALS, and it's so different from person to person. And how do we then create a cohort for clinical trials, especially if we want to find those early signals and really homogenize those clinical trial cohorts and test a drug? And we want to match people to the right trial based on disease biomarkers, genetic subtypes, etc. We want to understand the target engagement, as I mentioned earlier. Is the drug doing what it's supposed to do? If we think that a drug has to shake hands with a certain protein within the motor neurons of the brain or spinal cord, is it really shaking hands with the protein in people who are having the illness? And are we doing it with the right dosage before we bring it forward to the later phase testing? While we think about all the scientific aspects of the trial, what is really, really important is to improve access to clinical trials for patients. And we know that currently only about 10% of all people living with ALS participate in clinical trials or have an opportunity to participate in clinical trials. And this is very different compared to several cancer clinical trials where more than 90% of people who have a certain type of cancer participate in clinical trials. It's a completely opposite proportion that we're seeing in ALS, and we want to overcome the barrier of access to clinical trials, and we want this to be more accessible, and we want to bring these trials closer to patients' home as much as possible. And we all understand that drug development takes time and it takes money. And especially since emerging from the pandemic, research and development costs have really skyrocketed. And there are investment challenges. Many of the private investors and smaller biotech companies are actually struggling to find capital to focus on clinical development for ALS. And particularly when we think about the early phases of testing, that's where we run into more investment challenges. And how can we overcome that and make these trials affordable, not only for industry or biotech companies, but also many academic investigators who are translating drugs from bench to bedside, bringing them from preclinical studies into clinical studies. And there's also a huge effort for drug repurposing. There are many FDA-approved medications for other indications that potentially could have a mechanism that could benefit ALS, and Is there a way that we can create a platform where we can actually test those repurposed drugs in an efficient way and in an affordable manner? These are the challenges we're hoping to tackle through ALS MyMatch. To give you an idea of what ALS MyMatch looks like from a really high level schematic overview, starting from the left side, we start with this heterogeneous ALS population, and how do we then group them into homogeneous trial cohorts based on disease biomarkers? The entry point for an ALS individual is a common screening protocol in the ALS MyMatch program. And this common screening protocol has a very simple idea that can we use one set of screening data, medical history, medications, and a very targeted medical history and that's needed for a particular trial, a targeted list of medication supplements and other off labels that somebody might be taking that potentially could interfere with a particular clinical trial or could potentially be important for considering eligibility of a participant for a particular clinical trial. Can we collect one set of biofluid samples, blood samples, that we can then test for multiple different biomarkers that will tell us what would be a better match or a better fit for a certain participant based on the type of investigational product that we are testing in that particular trial. As we do this, this data the data that's generated in this common screening protocol would help us a lot in terms of reducing patients' participation burden in many ways. We know that sometimes participants will go through a screening process for a particular trial, and for whatever reason, if they don't meet all the criteria for that trial, they'll have to wait for several days to weeks to sometimes even months before they get an opportunity to participate in another clinical trial. Now here, and during that time, maybe the disease has changed a little bit, maybe the breathing scores have changed a little bit, or their blood numbers have changed. And so making it more challenging to find another trial where they're eligible and could participate in. So if we can maximize and optimize the utilization of the screening data and then try to find and match people to different trials that they could be eligible for. Let's say that a participant is eligible for more than one trial. Now, rather than having a computer decide and randomize a participant to a particular trial, we really want to empower patients to think of what might be a good trial for them to participate in. So here they have the option to decide, and either they can do it on their own or in discussion with their clinical neurologist to make a decision on what's a better fit for them to participate in and enter into that particular trial. And every trial is designed as an independent trial. Every trial may have very specific questions that we're trying to answer and may be very different from another trial. And so each trial will have its own design, set of outcomes, number of visits. And at the end, our hope is to get those answers, then to make a decision on whether that particular investigational product would move forward in a very efficient way into a later phase testing. Now, as we're doing the drug-focused clinical trial matching and data collection, we also want to contribute to furthering ALS research overall. Every participant that is entering the Healey ALS MyMatch program will also have their DNA banked for whole genome sequencing that'll be done after the trial is completed. Similarly, certain blood cells are banked at the entry of the clinical trial, from which those blood cells can be deprogrammed and then deprogrammed into stem cells and then reprogrammed into motor neuron cells that can grow in a dish. And if a trial is successful, our hope is to use those motor neuron cell lines to then look at what's called as a responder analysis. We know that in every clinical trial, there's a small subset of people who actually respond and others don't. We've never understood biologically what are some of the differences in terms of why some people respond better than others. And we want to use the opportunity of utilizing stem cell research and stem cell lines to understand responder analyses to the drug that was tested in that particular trial if a trial is positive. And so this is almost like a feed forward and a feedback loop. Feed forward in the sense, drugs that are being discovered from preclinical studies that are entering into clinical trials, and then we are learning about those in clinical trials. And then there's a feedback loop where we're also sort of then validating those learnings back into stem cell models and preclinical experiments, translational experiments, to better understand drug development and to really try to move the needle in a transformative way. Currently, the Healey ALS MyMatch program was launched from 4 sites. 2025, we launched at Mass General, Northwestern, and Chicago, Nova Southeastern Florida, and University of Minnesota. And we're hoping to expand the number of sites in 2026. And we're looking at site expansion currently. And all these sites are drawn from the NEALS Consortium. And all the sites that are part of Healey ALS MyMatch are also part of the Healey ALS platform trial as well. And these are all very experienced sites. And these are sites that have historically shown high enrollment numbers and they have the site staffing and bandwidth and capabilities to ramp up very quickly and enroll large numbers of participants in these trials. And as I mentioned before, the scope of ALS MyMatch is looking at this early phase and looking at several scientific questions as shown on the slide. And also, as I mentioned before, each trial is an independent trial, so every trial can have its own particular design, and it really opens up the opportunity for versatile trial designs. And these are meant to be smaller sample size clinical trials, anywhere between 20 to even up to 40, 60, or even 70 participants in each clinical trial, and it can be anywhere between 3 to 6 months long. Some of them can be open label studies and others can be randomized control trial. It could also have multiple arms if you're testing for different dosages that we're looking at. And some of them can have a blend of 1A, 1B, or going into 1B, 2A type of design in understanding and a spectrum of scientific questions at this stage of development. Now, I'd mentioned that one of our guiding principles for ALS MyMatch is really speed and efficiency. And how do we do this? There are lots of learnings from the Healey ALS platform trial that we have leveraged to build this program. And both our program teams at the Healey Center, the platform, and the MyMatch teams work very closely at the coordination center level, including all our IT systems, project management, quality assurance processes. We are working with the NEALS Consortium, and we're utilizing the NEALS trial infrastructure. We're very blessed to have an amazing set of patient advisors who are guiding us on making these trials patient-centric and making the whole program patient-centric as we develop this. It is still a young program, and we're very keen on bringing in innovations that would make it more patient-friendly and bringing trials closer to patients' homes. And we're utilizing NEALS also for reliance agreements that we have with each of the sites that are participating in the MyMatch program that allows us to use a single IRB and really move fast with those IRB approvals. And we have contracting processes that are fleshed out and really streamlined through the platform trial. We're using those processes. We're leveraging those processes even for MyMatch program. And we're using templated protocols, consent forms, all the source documentation that are used to gather data, and also electronic data capture system, case report forms, etc. All of those allow us to really have a short design phase and a rapid startup phase to move very quickly. And we have program level contracted vendors for various different operational aspects or even scientific biomarker aspects of the program as such. And so there are lots of time savings in ALS MyMatch. And as I mentioned before, time savings, we are very thoughtful about how we can save time at every step of the way in an ALS MyMatch clinical trial development. Starting from the design phase, can we be very efficient with the design of the trial itself? And typically the design phase can last for several months for a clinical trial. And here we have shortened the trial design time to 1 to 3 months, but it's it's a really tight run ship where we're working on a week by week basis in terms of deliverables and how we can get to that startup and activation phase. And the rapid site startup across all the participating sites also really shortens the overall timelines. And we have master agreements in place with all the participating sites. And once the sites are activated, as I mentioned before, all the sites that are participating in MyMatch are all handpicked sites who have committed to high enrollment for MyMatch trials, and we can really sort of hit the ground running with enrollment. And our hope is to really shorten referral to wait time, the wait times for participants from referral to trial screening. Oftentimes that becomes a bottleneck from the time that a patient expresses interest to be in a trial, or the clinician refers them to a particular trial, but to the time that they get a screening spot or a pre-screening spot can take several weeks. And we're trying to really shorten those wait times for patients. And I think having the common screening protocol, which is a standalone repository-like protocol, really sort of allows us to have those very short wait times for collecting that initial set of data that needed for trial entry. We're also we are putting in a lot of systems and processes in place that would bring a lot of flexibility for the initial screening procedures, including remote or e-consent that can be done virtually, even if a participant lives around 100 miles away from a study center. And the screening window itself is quite broad. That allows for screening procedures to be done over multiple days. There's a lot of flexibility. We've also simplified things to an extent that can be incorporated within clinic workflows. If a particular clinic prefers to, or a participant who's living far away prefers to integrate a research visit on the same day as their multidisciplinary ALS clinic visit. And many of our trials are utilizing local labs, local safety labs that are CLIA certified safety labs. That makes it, again, very convenient for patients rather than having to travel hundreds of miles to go to a study site only to get a blood drawn to look at their liver tests or kidney tests. They can go to a local lab and get these drawn and can be utilized for trial purposes and for analyses. And so that's during the enrollment and the follow-up period of the MyMatch trial. And then even from the time that the last participant completes their last visit in a MyMatch trial, our closeout plan, gathering the data, cleaning up all the data from that time, from the last participant's last visit, and moving up to the database lock and analyses and reporting of the trial results. So we're hoping to compress timelines end to end, starting from design all the way to reporting of the results in a MyMatch trial. And to give you an example of what we have seen so far, we launched the first trial, the Acacia trial, earlier this spring, and we launched this trial in less than 3 months from the time that we started working on the MyMatch program. Typically, clinical trials take 6 to 9 months for startup, and so we worked under very, very aggressive timelines. And our first participant's first visit was in May, end of May. And in just a little over 4 months, the trial was completely enrolled. And we enrolled 41 participants across 3 sites. And I've highlighted on this slide in terms of our enrollment rate. Typically, sites enroll at the rate of anywhere between 0.5 participants per site per month. That has been the historical average. To even the high enrolling sites will enroll at around 1 participant per site per month. And as you can see here, many of our sites enrolled at 5 to 6-fold higher enrollment rates than historical averages, which is super exciting to see. It's really about pushing boundaries and seeing what we can achieve in terms of really moving in a direction where we can ramp up access and enrollment timelines to as high numbers as possible. And even in terms of startup timelines, our IRB approval timelines were under a month, which is also historically, compared to historical averages, very, very short. Site contracts were also executed in a very efficient manner. In under 1 month, all 3 site contracts were executed. So this is the first trial that we launched, and it is super exciting. And the question is, you may be wondering, what is this Acacia trial and what are we testing here? Acacia trial is testing a repurposed drug called digoxin. And this is a really beautiful example of bench to bedside translation that happened in a very fast and efficient manner. It's all work from one of our colleagues, Brian Wenger, and their lab, Christine Marquez from Dr. Wenger's lab, did series of experiments on digoxin, which is a cardiac glycoside, it's a cardiac medication. They did 2 independent drug screens. They looked at over almost close to 2,000 different FDA-approved drugs, and they looked at 2 different models. One is a C9 model, and the other is a sporadic ALS or TDP-43 model, and they looked at stem cell models. And they looked at what actually improves survival of the motor neurons cell lines, as well as what improves the neuritic growth in both of these models. And in both of these independent drug screens, cardiac glycosides emerged as a class of drugs that improved survival and neuritic cell growth in both of these models. And there are several cardiac glycosides, among which digoxin is the well-known. It has been around for almost half a century now. It has been used in heart failure and for cardiac indications. And knowing that digoxin or the cardiac glycosides is a class of drugs that emerge on both of these screens, they did a second set of independent experiments looking at TDP-43, which is the protein that aggregates in ALS, and how the biology of the TDP-43 can be reduced or reversed. And in a second set of experiments, it did show that there was benefit in those stem cell lines. And based on this, the preliminary data that is so exciting, this medication, digoxin, which is FDA approved for a cardiac indication, was brought into a clinical trial. And there are 2 separate cohorts in this clinical trial, one that's focusing on early ALS, sporadic ALS individuals, and another that's focusing on the C9 genetic subtype of ALS. And this trial was unique in many ways. And many trials will have 1 or 2 dosages that we would test. But in this particular trial, we really wanted to sort of titrate this and personalize it based on what each individual may need to achieve a certain blood level of digoxin. So that was one of the most challenging aspects of running this trial, is that each individual's digoxin dosage had to be titrated almost on a weekly basis until they achieve a certain range of digoxin blood levels that is within certain parameters of safety, as well as what's known in cardiac indications as being beneficial. We don't know if that's the same dosage range that would be beneficial, that would enter the brain and the motor neuron cells in ALS, but that was our starting point, and that was sort of our guidepost for the Acacia trial. Sorry, did somebody have a question or?
Yeah, it's just some side talk. We'll take care of that. Go ahead, Suma.
Okay. And so in addition to safety and tolerability, we looked at several biomarkers in this trial, including neurofilament, and we were looking at whether the drug enters the blood-brain barrier and what the CSF levels are. This trial has not completed yet. We've completed the enrollment, and we are anticipating to have results from this trial sometime early next summer. And so far, that was just the first trial that was launched this year. And we're super excited and really encouraged by the preliminary trend that we're seeing in terms of the participation of sites and the interest that the drug developers, biotech companies, and academic investigators are showing in the ALS MyMatch program. Is super exciting. And so far, 38 companies have expressed interest to partner with us for ALS MyMatch. Earlier this spring, we had a request for application to participate in MyMatch. We received 12 applications that were reviewed by an independent therapy evaluation committee that vets these applications for the drug, the target, the pathway, the fit, the relevance, and potential impact of these investigation products. And out of those, 4 were selected for 2025-26 cycle. and two additional ones are under scientific review, and we are accepting applications on a rolling basis. And the first trial was the Acacia trial, as I mentioned to you before, and the two other companies, Tiziana and Inflammasome, are under a design contract for ALS MyMatch. We're developing a protocol for both of these, and once they complete this, then the next phase would be to submit it to FDA to get may proceed, and then go into the operations and open it up for enrollment. So we are working as hard and as fast as we can to bring this to patients as soon as possible. And then we have two additional companies that are currently under contract negotiations, and we're hoping for an early 2026 design launch for two additional compounds. So if you're wondering, how can I become a participant in ALS MyMatch program, there are 3 steps. The first step is to understand sort of the preliminary eligibility criteria for MyMatch. So all participants are required to have a diagnosis of ALS and have weakness less than 24 months from symptom onset, and have breathing scores that are at or greater than 65%. And all ALS participants are required to enter the program with ALS genetic panel testing, which is routinely done as standard of care for ALS in the US. And once you meet the initial preliminary eligibility criteria, the second step is to complete the common screening protocol, where more detailed screening information and comprehensive information and blood sample is collected for biomarker testing. And depending on what we learn from the common screening protocol, the next step is to decide if you're eligible for more than one trial that may be enrolling at any given point in time, what would be a best fit for a participant to enter a clinical trial. So what goes on behind the scenes to build a program like this and launch at multiple sites? We have a large team at the Healey Center, at the NCRI, who are just an amazing team to work with and learn from on a daily basis. And every day is a new day with new innovations and ideas that comes from every single member of our coordination center team. And we have amazing leaders and mentors, including Dr. Mary Sukovich and Dr. James Varian, Sabrina Paganoni, who've been supporting the ALS MyMatch program. And it is just wonderful to see all of this come together. And with that, I'll leave you with some information about the Healey ALS MyMatch as a website, and we are constantly updating content on this on our website about MyMatch and upcoming trials and enrollment links. And we also have the ALS link where you can get regular updates through email e-blast that will be sent out about. It's not just about MyMatch, but all information that are disseminated from the Healey Center. There are a few webinars that we have already conducted, and we're hoping to have a regular cadence of community webinars on our Thursday afternoon webinars hosted by the Healey Center, and we'll have more information for you on the cadence for the MyMatch webinars. And if anybody's interested in learning more about MyMatch or any of the trials, or if you're interested in participating, there's an email address. Please feel free to send us an email. And as I mentioned before, we are accepting applications also from drug developers, pharma companies, and biotech companies, if anybody's interested in partnering with us. With that, I will stop sharing and open to taking any questions.
Thank you so much, Suma, for this insightful, and not only that, it's such an innovative way. And, at MGB, and you guys keep trying to make sure that, the trials are faster, cheaper, better, with less placebo impact, because things we can't change with FDA, you guys are trying to change it for the patient. So I so appreciate it. And we have a lot of questions that came in chat, and I'm going to invite my colleagues here, Anusha and Alex, to come in and do the moderating of the questions. And also, before Anusha and Alex, you start, just do a 10-second background on you guys. It's both of us are our student fellows, and I would love to introduce both of you.
Thank you so much, Indu. Yeah. So first of all, Dr. Babu, thank you so much. My name is Anusha. My uncle has ALS. He's had this disease for 10 years now. And when he was diagnosed, I was so, impacted by this disease. And so it's been something that is really close to my heart. So I've been working with the foundation for about 2 years. I was an AI engineer. I recently graduated from Hopkins, and now I'm pursuing med school. So that's what I've been up to. But yeah, Alex, do you want to go ahead?
Yeah, hi everyone. My name is Alexandra, and thank you, Dr. Babu. That was amazing, and we're really grateful to have you here. I've also been working with Everything ALS for the past 2 months, and Dr. Ostrow at the ALS clinic at Temple actually introduced me to the organization. I currently work as a medical scribe at Weill Cornell Medicine for an ENT doctor, and I also am applying to medical schools currently.
All right, great. So we can go ahead and get started with the first question. So the first one is, what biomarkers do you specifically use to identify the efficacy of new drugs? Is it only neurofilament light chain? And then how do you subgroup patients straight away and kind of on what principles? I know you kind of briefly talked about that, but maybe could go in more detail.
Absolutely. I don't have one single answer because every trial is going to be different. It all depends on the drug and it depends on the pathway. And there could be drug mechanism specific biomarkers we may be looking at. We may be looking at neurofilament, as you mentioned. We may be looking at genotypes. Maybe there are some drugs that work better in certain genetic subtypes. And some of the trials will also include imaging biomarkers. So we have an imaging advisory panel that guides us on optimal use of biomarkers, both for entry as well as to consider as outcomes or readouts.
Thank you. Our next question is, can I use right to try if I can't join the trial?
That's a very interesting question. So, right to try, expanded access, and clinical trials are slightly different pathways to get access to investigational products. Now, what is being tested in MyMatch could include experimental drugs or could also include repurposed drugs. And some of the repurposed drugs are available as FDA-approved medications that potentially your clinical neurologist, if they're comfortable, and you could request for access as an off-label. Now, in right to try is a slightly different pathway where one would have to work with their clinician and try to get access to an experimental medication. And that is not inherently tied to the MyMatch program itself, but that is something that you will have to work with your clinical neurologist and talk to your doctor to see if that's the right drug for you, that's the right pathway, and then work with the drug developers as well.
Thank you. Next question is, are any of the new sites going to be on the West Coast?
We are looking at site expansion. So we're using there's a process and a methodology, and we're hoping to expand sites, and we're being mindful of geographic location of sites. We also want to make sure that because we're building this program on speed and efficiency, sites that we're selecting are able to commit to high enrollment numbers because we do want to improve access for all our trial participants.
This is also going off of that a little bit, but what is the possibility of opening a site or sites in Canada?
Currently, we are looking, that's an excellent question. Currently, we are looking at sites in the US only. It's more for regulatory reasons more than anything else.
So the next question is, it seems like MyMatch almost feeds people into the right clinical trial. And so how do you think we can then solve this next issue where a lot of the patients don't even qualify for a clinical trial because of maybe the time due to onset or their breathing scores or kind of really strict eligibility criteria that make it less accessible?
That's an excellent question. As a field, I think we'll have to address this question at every step of drug development. We are encouraging drug developers or companies to think about a flexible cohort where we are looking at matching people to drug and biology and the primary analyses data set that we're looking at to answer those scientific questions. We're also encouraging companies to think of another cohort where it's less restrictive in terms of eligibility. More to come on that. But not every trial will have the same set of eligibility criteria. For clinical trial testing, the field is, I acknowledge that the field is moving towards enriching trial cohorts with earlier disease. And this is not with the intent of excluding people from getting access to a drug, but primarily to see if we can strengthen the possibility of finding a signal in smaller cohorts so we can move the drug development quickly along the pipeline, because ultimately our goal is to get to that FDA approval so that becomes available to thousands of patients widely as a standard of care medication. And how can we do that in an efficient way? And at the time that we're testing these drugs in clinical trials, we don't know which ones would work, which ones don't. And there's this concept of what's called as, we call it as clinical equipoise. We don't know. We think that they all probably work and they all probably have an effect. We don't know which ones actually will have an effect and at what stage of the illness as well. We go with some preclinical information that guides us in terms of how to test this. Now, for people who don't qualify for clinical trials based on the disease duration or breathing scores, etc., there is an alternate pathway called Expanded Access Program. And even compared to the last 5 years, the Expanded Access or EAPs have grown so much in terms of the availability, the numbers. In 2018, when we first started talking about EAPs and we enrolled the first participant in an EAP, we had no idea how quickly this is going to grow. And now we have large, even NIH-funded EAP programs that are bringing access to a few hundreds, and now cumulatively it may all add up across multiple different programs. It's still not enough. There is still a huge demand-supply mismatch, but we're heading in the right direction, and that's the most important thing.
Thank you. Will asymptomatic gene carriers of ALS be eligible for enrollment into MyMatch?
That is such a fantastic question. Our hope is, our hope is that eventually, when we start sort of thinking about drug development and designs and cohort enrichment, we want to move to a phase where we are designing trials with earlier and earlier intervention, and that goes even into this asymptomatic at-risk population. But we need more. Again, we're all thinking about, and the field is heading in that direction. We already have the first pre-symptomatic at-risk clinical trial for SOD1 ALS. It's just a matter of time before we start expanding into others. So at the moment, MyMatch is available for people who are symptomatic with ALS, but our hope is to head in that direction.
Awesome, thank you. This next question is a bit of a clinical question. So someone said, I understand that digoxin can be useful, but it can also come with a few side effects as it's difficult to titrate the dose. I'm also curious if it could be more dangerous to use in patients over 50.
Oh, that's an excellent question you ask. And like any medication, digoxin has side effects. Like with any medication, we'll have to have good safety monitoring parameters. Within the clinical trial paradigm, we have a safety review committee, and there is a lot of stringent oversight on data that's coming in from clinical trial participants. We're keeping an eye on their blood levels, the EKG parameters, everything that gets entered into the electronic data capture system. Now, outside of the clinical trial at the moment, in ALS population, we don't know everything about the safety. We don't know everything about the tolerability issues. But outside of ALS, there's a ton of data about digoxin and its safety profile and tolerability profile. It does have side effects, and those side effects tend to be, it's not dangerous, but it tends to occur more frequently if the blood levels of digoxin is above a certain threshold. And one of the things we're doing in the clinical trial is to make sure that everybody's digoxin blood levels are staying within the range of safety parameters that are set by the cardiac field. And I think this trial, we'll learn a lot from this trial in terms of ALS population and what is the well-tolerated range of dosages and the safety profile. But as such, because we have tens of thousands of patients, individuals who have taken digoxin over the last half century or so, we know exactly what to expect in terms of dose-dependent toxicity, and we want to stay away from that. And we do have people above 50 in over 50s who are taking the medication in the trial, and they're doing okay.
Thank you. Is there a projected % increase in the number of trials of experimental new treatments that MyMatch will result in?
Increase, can you repeat that question one more time? Percentage?
Yeah. Is there a projected % increase in the number of trials of experimental new treatments that MyMatch will result in, if there is?
I don't have a percentage increase, but we've built this infrastructure in a way that can support multiple clinical trials to run at the same time or sequentially. And we're building this infrastructure in a way that we can actually expand the number of sites and sustain this program over a long period of time. And we hope that there will be multiple clinical trials and multiple drugs that will be tested in this program. The match paradigm actually is not new to the field of medicine. Oncology has run several match clinical trials over the last decade, and some of them have been hugely successful. And many of them have been supported by NIH, the National Cancer Institute. And when there's large federal funding and support for a large clinical trials network, it really increases the scale and scope of running such trials. And in fact, I think some of the match trials have even run up to 37 or 38 different clinical trials. So it's all about how do we expand the infrastructure, and MyMatch so far has been built purely out of philanthropic support. It was a matching challenge grant we got from a family foundation, and we were fortunate to launch the program and allow for some cost sharing for initial companies that were accepted into MyMatch. But we want to grow and allow for multiple trials to be a part of MyMatch.
Awesome, thank you. I think a big concern for people is the 24-month disease onset rule, and if people kind of fall outside of that. And so in your opinion, what do you think needs to happen in order to make that more flexible and be able to kind of apply the super innovative approach to a bigger population?
Absolutely. And so the 24-month cutoff is again having some sort of a guide to enroll earlier population in these small trial cohorts. As I mentioned before, these trial cohorts are small, right? Ranging between 20 to 70 or so participants. And when you have a very heterogeneous population and we put them all in one bucket and try to test a drug, we may dilute the signal. And even if there is an effective drug, we may lose that signal, and potentially we may call it as a false, it could be a negative trial and it could be a false negative, and may not even move forward to the next phase. So if there's a way to sort of homogenize trial cohorts and really intervene early where the disease biology is amenable for disease modification or change in trajectories, that's our hope, and that's where the 24 months comes into picture. Our hope is that in these short and fast clinical trials, once we know these answers, we move into the next phase of study. And there, our hope is that the field will move towards having two pathways for people in the clinical trial pathway, where we can test clinical efficacy, and an expanded access program as a parallel pathway for people who don't qualify for clinical trials, so they can still get access to the experimental drug and also generate data from the expanded access. We've run a number of EAPs where we know they're so valuable in generating real world data about safety as well as biomarker data.
Thank you. If someone has already participated in a clinical trial and completed all of the prerequisite tests for MyMatch, could their clinical trial test be used to apply?
No, any previously completed clinical trial data will be a part of that particular clinical trial. The data that is generated in the common screening protocol after you consent for the MyMatch clinical trial common screening protocol, that's the data that could be used for any of the MyMatch trials. And there is a window. The common screening protocol has about a 45-day window. And so many of the, especially safety labs, et cetera, that are collected during that window would be relevant only for that duration of time. It doesn't mean that if somebody couldn't get into a trial in that 45-day window, they won't be eligible for MyMatch trials moving forward. They just have to then a lot of the information collected from the previous 45-day window can be pulled into their new screening data, but they may have to update some blood work or some breathing scores, etc. So a few things may need to be repeated.
Thank you. So given that because MyMatch kind of focuses on people within the 24-month onset, patients within that window can qualify for both the early phase trials like phase 1 and phase 2, and then more later phase options. So how do you think about it when it makes sense for someone to kind of join either of those, and kind of what considerations do you think matter the most?
That's an excellent question. Within MyMatch, all of the trials are early phase clinical trials, but if somebody is within 24 months, potentially they could qualify for other later phase testing programs as well. It really boils down to sometimes, as I was mentioning, that there is clinical equipoise. We don't know which of these work ultimately, and which of these experimental therapies potentially might move towards that FDA approval. But ultimately, it boils down to what's a better fit from a logistical standpoint? Maybe one requires somebody to come in on a weekly basis, or they have long study procedures, and it may not be feasible for an ALS participant's lifestyle or work schedule, or even distance from the site, etc. And whatever might be more convenient and logistically feasible, that may become the right choice. Or if there is one that is dedicated for a certain genetic subtype, and they carry the genetic subtype, then potentially that might be a good fit as well.
Thank you. Our next question is, can you talk about the Koya MyMatch, which is open for enrollment soon?
Koya is a separate clinical trial. It is not a part of the MyMatch clinical trial, and they're testing a drug that targets IL-2 pathway, which is a neuroinflammatory immune pathway, and it is not a part of MyMatch. It's a separate clinical trial altogether.
So this is a question from someone who said that, I know this is separate from MyMatch, but I've been hearing that Neurology might be joining the Healey platform trial in Q1. So could you share more insight into this treatment in the trial? And I'm particularly interested in how it fits into the broader Healey system.
Thank you for that question. So I am not directly involved in the Neurology clinical trial. I know that there will be a webinar on our Healey Center Thursday webinars, and there will be an upcoming webinar that talks about the drug, the science, the mechanism of action of the drug, and the eligibility criteria, et cetera, for that particular trial. So maybe if we can put a pin on that for now, and then happy to share more information through the Healey webinar series.
Thank you. Can you talk about what is being studied in the trials coming up with Tiziana and inflammasome?
Absolutely. I can give you a high-level overview. You've probably seen press releases about both of these. Both in Tiziana as well as inflammasome are under design contracts for MyMatch. Tiziana has a compound called foralumab, which is a nasal spray of a monoclonal antibody that targets an inflammatory immune pathway through something called as an anti-CD3, which follows the regulatory T cells or Treg pathway. Tregs are an important aspect of ALS disease biology. There are various different approaches of testing medications, or there are cell-based approaches, monoclonal antibody approaches, and immune modulator approaches that are all sort of targeting the regulatory T cell pathway. And this one is a very patient-friendly drug delivery approach through a nasal spray. It doesn't involve infusions and invasive procedures as such. And so we'll be using biomarkers to test whether the drug enters the central nervous system and engages with the target, and it helps with neuroprotection. So more to come on the design of that trial. Inflammasome, on the other hand, has a drug called camivudin, or for short called as K9. And this also targets an inflammatory process. And it is currently being tested for ocular indications that also involve the same sort of inflammatory proteins. And through this, after the MyMatch trial, we are in the design contract with the company, and we're working on developing the trial design and optimizing what biomarkers we will be using for both entry as well as for readout of the MyMatch trial. And in the coming months, we are anticipating to have a webinar through, again, the Healey webinar series. We'll be presenting about both Tiziana as well as inflammasome and the drug and the mechanism of action and the target.
We had a bunch of questions about how people can access expanded access programs. And so maybe you could explain a little bit more about how they work and kind of how people can think about that.
Absolutely. Expanded access programs is a separate pathway through the FDA as a regulatory pathway for any life-threatening illness where there is an experimental therapy that potentially could have a benefit for that particular indication. And EAPs can be of various different kinds. And sometimes an EAP can be developed and designed as a single patient EAP, where a patient would work with their clinical neurologist, and if it is an appropriate fit to test a certain experimental medication, they would reach out to the company that's manufacturing the drug and request for a single patient access to the experimental drug. And it is a process. Then you would have to write a protocol, submit it to the FDA as a single patient IND, and then get access to it. Some of the antisense oligonucleotides, like even tofersen for that matter, before Qalsody or tofersen, before it got approved, became available as a single patient IND EAP after the pivotal trial had completed, and there was some preliminary data suggesting that it could be benefit. Many times that becomes that early access for that can be customized or personalized for an individual patient. Sometimes EAPs can be designed as a cohort or an intermediate IND program. Many of the Act for ALS funded EAPs are considered as intermediate or cohort-based EAPs. And these cohorts can range anywhere from 5 participants to up to 200, 300, 400 participants. And many of the NIH-funded EAPs actually are targeting large cohorts of people. It can be at a single center, it could be at multiple different centers. All NIH-funded programs are multi-site EAPs. The large multi-site expanded access programs are usually posted on ct.gov. So when there are new EAPs that are enrolling, you can always look at ct.gov for information around currently enrolling EAPs. One of the EAPs that is NIH funded is the ibutolast EAP that is currently enrolling at certain sites, so you can get information about that. And from time to time, there may be other EAPs that companies may also sponsor, or a certain academic center may be offering to participants in very small numbers as well. So talk to your neurologist as a first step to see if you qualify for an EAP. Generally speaking, EAP there's an FDA requirement that people who are accessing EAPs should not be trial eligible. The idea is our fundamental focus is still to develop drugs that potentially can go towards that FDA approval and become standard of care and become available to thousands of patients who have ALS. And we don't want compassionate use or expanded access programs to jeopardize that drug development pathway itself. And so, in order to qualify for EAP, there's a requirement to show that you don't qualify for other clinical trials as such.
Thank you. Could you share how many drugs are expected to be tested over the next 24 months and when we might hope to see interim updates?
In MyMatch specifically, I'm assuming.
Yes.
In early next year, we're hoping to launch, as I mentioned before, at least 2 drugs that we're currently in design, and potentially 2 others. As soon as the contracts are finalized, we'll be launching those as well. So in 2026, we're hoping to have at least anywhere between 2 to 4 that we'll be launching. Could be more as well. It all depends on how many applications go through the vetting process of the therapy evaluation committee and get selected to be a part of MyMatch. It's very, very exciting. There's so much interest, and and the disease biology, the drug targets and biology that we're seeing now is just it's really incredible. And many of them have very targeted pathways and biomarkers, and the way the field is really heading right now in ALS is there's a lot of hope.
Thank you.
All right, Dr. Babu, I have one last question for you. So your work is really focused on making trials more efficient. And so I wanted to ask, how do you think an AI-powered clinical matching tool like EverythingALS's Sava AI will assist in making MyMatch run more efficiently?
This is such an important and very, very exciting question. And Indu and I have had many conversations about this. We're really hopeful that utilizing AI for pre-screening, for matching, for identifying potential participants would actually be so wonderful to partner with and really expand access to clinical trials and the patient community. So more to come on that in the coming months even.
All right, thank you so much.
Oops.
Thank you so much, Dr. Babu. I wanted to tell you that Anusha's fingerprints are also all over Sava AI. And a lot of the questions today about how do I access EAP, how do I, get to know faster, all that is really bringing in Sava AI. I just wanted to let the community know Dr. Babu has worked very, very hard in helping us make sure that we can actually match the patients faster and get to, the trial enrollment faster. So we are very thankful for all the insights you've shared, and you continue to work with us on making that happen.
EverythingALS is doing amazing with all the innovation and technology. It's fabulous.
Thank you.
Dr. Babu, how did you like the audience's questions? Did you think that we are doing our homework?
This is such an amazing audience. I'm very, very thankful for this opportunity to present today. And all your questions were so thoughtful and insightful. I really appreciate it. And this is really the type of collaboration, partnership, and community partnership that we love to continue to grow and really bring the ALS community together. Everything ALS and all of you are unstoppable. And at the Healey Center, we are all unstoppable as well. So I think together we have a mission that we're working together towards.
And this is the community that we at EverythingALS is trying to provide for everyone.
Thank you. Thank you for doing that.
Well, we're on your team. So anytime you need some practical information, you know where to come and get it.
It's amazing. We're one big team, and it's so lovely to see that.
Thank you so much, Dr. Suma Babu, and thank you for your time and your evening spent with all of us. And now we actually go into our open forum where people discuss any topic that works. So, but don't want to take any more of your time and your effort as you're working very hard to make it easy for us to get into the trial and make some of the trials successful. Thank you so much for all your time effort and your energy in for us and ALS community.
Thank you so much for having me. Really appreciate it. Thank you.
Thank you.